Now enzymes. So let us see some of the important questions related to the chapter enzymes. Now let us see the first question. Enzymes belong to which class of compounds? Options A, polysaccharides, B, polypeptides, C, polynitro-heterocyclic compounds, D, hydrocarbons. So here enzymes are considered as proteins, or we can say all enzymes are proteinaceous in their nature. That is here they proteinaceous in the nature means they are belonging to the class that is polypeptides. Why? Because here all the proteins are made up of polypeptides only, and polypeptides are made up of amino acids. That is what, that is the relation that is existing between the enzymes and the proteinaceous nature related to the enzymes. So from the given options, option B, that is B, polypeptides is the correct option. Question number 2. The enzyme secreted by digestive glands of Nepenthes is options A, pepsin, B, amylase, C, urease, D, catalase. So here Nepenthes is an insectivorous plant which is growing in nitrogen deficient soils. Here it is showing a specific type of modification in its leaves. So it contains the modified leaves, or we can call such type of leaves as a trap leaves. The leaves are modified to trap the insects. So what is the need of the plant to trap the insect means, so as it is growing in the nitrogen deficient soils, so it had to compensate the deficiency of nitrogen. When they are growing nitrogen deficient soils, they will trap the insects, and that whenever the insects are trapped, so the proteolytic enzymes which are present in them will act on the protein part of the protein part present in the insects, and they will digest them. So that means the digestive juices which are produced by them, that is they have the digestive glands. The digestive glands will secrete the juices called as a digestive juices. So the digestive juices or the digestive glands which are producing these digestive juices will contain pepsin enzyme. This pepsin enzyme will act on the proteinaceous substances present in the insect which have been trapped, and they will break the bonds, peptide bonds, and they will convert, they will simplify the protein structure into amino acids, and they will absorb the nitrogenous substances. That is, they are compensating the deficiency of nitrogen present in the soil. That is the reason why we can say that the digestive glands of Nepenthes will contain pepsin, which is required for the breakdown of protein molecules. So from the given options, option A, that is pepsin is the correct option. Question number 3. Inhibition of enzyme action due to accumulation of end product is called. Options A, competitive inhibition, B, non-competitive inhibition, C, feedback, D, metabolic inhibition. So here first one, competitive inhibition. So what do you mean by this competitive inhibition means? So this is one of the method of inhibition of enzyme action, where the inhibitor will have the similar structure as like that of the substrate molecule, and before the substrate go and fit into the active site, this inhibitor will go and occupy the active site on the enzyme, such that the activity of the enzyme will be inhibited. Second one, non-competitive inhibitor. Here non-competitive inhibition means the inhibitor does not have similar structure as like that of the substrate molecule, and it will not fit into the active site of the enzyme because it is not having the similar structure as like that of the substrate molecule. But the non-competitive inhibitor will fit into any part of the enzyme other than the active site, and whenever it is attacking any other part, it will induce the enzyme such that the active site, the shape and structure of the active site will be changed. That is the way how these non-competitive inhibitors are inhibiting the enzyme action. The third one is the feedback inhibition or feedback process. So here the feedback inhibition means the end products or the products which are formed in a process or in a chemical process, they will get accumulated. So because of the accumulation of the end products, the accumulated end products will show its effect on the enzyme which is required for the first reaction. So here this is because of the accumulation of end products. And the fourth one, that is metabolic inhibition. So that is the inhibition process which is related to the function or process will get inhibited. So here from the given options we can say that more related option is the accumulation of the product is nothing but the feedback inhibition. So from the given options, option C is the correct answer. Question number 4. Proteins giving rise to peptides. Enzymes catalyze the above reaction belongs to this class. Options are A 4, B 2, C 5 and D 3. So here proteins are converted into peptides. So here there are 6 classes of enzymes are there. They are depending upon the type of function that is performed by those enzymes. So here there are 6 classes, class 1, 2, 3, 4, 5 and 6. So here the breakdown of the peptide bonds present between the amino acids of the protein will be taking place here with the help of some enzymes like pepsin like enzymes, and they are converting the long chain of proteins into small chain of polypeptides. So here that is done by the group of enzymes which belong to the group 3, that is peptidases. So from the given options, option D, that is class 3 is a correct option. Question number 5. Enzymes catalyzing substrate level phosphorylation reaction belongs to. Options A oxidoreductases, B phosphatases, C transaminases, D transphosphorylases. So here the question is related to the substrate level phosphorylation. Here substrate level phosphorylation means the formation of ATP molecule directly involving the substrate molecule without the electron transport system. This type of process can be, or ATP synthesis can be seen in both glycolysis and also in Krebs cycle of respiration process. So here in this one what is happening is the substrate plus the ADP molecule will be taking part and leading to the formation of the product along with the ATP molecule. That is substrate will be losing one phosphate which is accepted by ADP, and the substrate is converted into product, and ADP is, as it is accepting one more phosphate, the diphosphate will be converted into triphosphate, that is adenosine triphosphate. So what is happening here, it is the transfer of phosphate group from substrate molecule to the one substrate molecule to the other substrate molecule is taking place. So leading to the formation of ATP, that is a phosphorylation. So that's what we can say that this is a transphosphorylases. So which are helping in the, or we can say that the transfer of phosphate group from one substrate to the other substrate molecule is taking place. So that's what is that is the reaction that is taking place in the substrate level phosphorylation process. So from the given options, option D is the correct answer. Question number 6. Enzyme coded by enzyme code 2712 in modern classification the given options: A. Hexokinase, B. Aldolase, C. Enolase, and D. Glucose 6-phosphotransferase. So here every enzyme will be given the enzyme code, that is what we call it as EC. So here in the given question 2712 is given. So here based on the classification, every enzyme will be placed in the classes, that is 6 major classes are there, and in that class depending upon the other characteristic features, they are grouped into subclasses. The first one is the class, and under each class the subclasses are present, and under each subclass subclasses are present. And here under subclass every enzyme is specified with some number. So in the enzyme code 4 digits have been given. The first digit is related to class, second digit is related to subclass, and third digit is related to subclass, and the fourth digit is related to the serial number or sequence number of the enzyme present in the subclass. So based on that one, in the question it is given as 2712, that is first digit 2 is related to the class, second digit 7 is related to subclass, third digit 1 is related to subclass, and fourth digit 2 is related to the serial or sequence number of the enzyme present in that particular subclass. So here in the given options, first one, that is hexokinase have been given. Hexokinase is the one which is belonging to transferases, where the we can say that in that subclass is the phosphotransferases, that is a transfer of phosphate group, we can say, from one substrate to the other substrate molecule. Whereas second one, aldolases. Aldolase is the one which is required for splitting of the molecule in absence of water. And third one, that is enolases, that is related to the water molecule aspect. And the fourth one, that is D, glucose 6-phosphotransferase. So here it is a phosphotransferase, that is it is belonging to the second class, that is called as transferases. Under the transferase, that is second class, that is 2. Under transferases it is belonging to the 7th subclass, where the phosphate group is transferred, and under the subclass it is belong to subclass where the alcohol substitution can be done, and the last digit 2 is related to a serial number of that particular enzyme in that subclass. So from the given options, the enzyme code 2712 is represented by glucose 6-phosphotransferase. So from the given options, option D is the correct answer. Question number 7. Enzymes which are associated with the formation of new bonds using energy from ATP hydrolysis are. The given options are A. Lyases, B. Ligases, C. Kinases, and D. Reductases. So here the question is related to the formation of new bonds. The formation of new bonds, that is using energy. So using energy in the form of ATP molecule. So here how does ATP molecule is provided means, when ATP molecule will undergo hydrolysis, the bonds will be broken, that is it is converted ADP and inorganic phosphate. So that is here the third, or we can say that the highly energy containing bond present between the ADP and the third phosphate will be broken to release 7.6 kilocalories of energy. So that is the way how the hydrolysis is yielding the energy. And here in the given options, the ligases are the one which are useful in the formation of new bonds using the energy, that is ATP molecules. So from the given options, option B is a correct answer. Question number 8. Glucose 6-phosphate plus ADP the enzyme which catalyzes the above reaction belongs to the class. So, here options are A 1, B 3, C 4 and D 2. So, here actually glucose 6 phosphate plus ADP. So, for this process what happening is the transfer of the phosphate molecule is taking place. So, here the transfer of phosphate molecule from one substrate molecule to other substrate molecule, where the glucose 6 phosphate is accepting or taking place the transfer of the phosphate molecule. That is what we call it as a transferases. That is particularly in that class transferases, the phosphotransferases is the one, that is kinases we can call them as. So, here the enzyme which is participating in this reaction will belong to the class called as transferases. So, from the given options, option D, that is class 2 is a correct answer. Question number 9, identify the incorrect regarding enzymes. The given options: A, aldolase acts on different types of substrates. B, during reaction enzyme shows neither quantitative nor qualitative changes. C, enzyme never changes the equilibrium of an reaction. And D, all enzymes of a cell have same optimum pH value. So, here in this question we have to identify the incorrect statement regarding enzyme molecules. So, here option A, the aldolase is the enzyme which is taking part in conversion of fructose 1,6-bisphosphate to DHAP, that is dihydroxyacetone phosphate, and G3P, glyceraldehyde 3-phosphate. Here in this reaction aldolase is taking part, and here the substrate is fructose 1,6-bisphosphate. At the same time aldolase can act on the reverse reaction also, where it will the reaction in which DHAP plus G3P giving rise to fructose 1,6-bisphosphate. Here we can say that in the first reaction the fructose 1,6-bisphosphate is substrate, whereas in the second reaction the same enzyme is taking part, but the substrates are GHAP and G3P. So, aldolase will act on different substrates. That statement, that is correct. And statement 2, during reaction enzyme shows neither quantitative nor qualitative change. So, that is correct, because when an enzyme enters into reaction it will try to show its effect on the modification of the substrate molecule such that the substrate will be converted into product, and whatever the nature of the enzyme which is entering into the reaction, the same type of enzyme will be coming out of the reaction. That is there will be change in the substrate molecule, but not in the qualitative nor the quantitative change in the enzyme molecule. So, this statement is also correct. Now, let us come to third one, that is enzyme never changes the equilibrium of an reaction. Yes, if a reaction is taking place in absence of enzyme, whatever the equilibrium that is present between substrates and products, the same type of equilibrium will be maintained when the same reaction is taking place in presence of enzymes also. That is, no change in the equilibrium of the reaction. And option D. So, here all enzymes of a cell have same optimum pH. So, actually different enzymes will have their activity at certain levels of pH molecule. So, the some of the enzymes are active at 2 pH, or some of them are active at 5 pH, some of them are active at neutral pH, that is 7, and some of them are active at alkaline pH also. So, that means we cannot say that all enzymes present in the cell have the optimum pH value. The pH values will be different for different types of enzymes which may be present in the same cell also. So, here of all the options that have been given, this is a correct statement, second one is correct statement, third one correct statement, whereas the fourth statement is a wrong one, because all enzymes which are present in a cell will have different optimum pH values. So, from the given options, option D is the correct answer. Question number 10. An example of competitive inhibition of an enzyme is the inhibition of options: A. Succinic dehydrogenase by malonic acid, B. Cytochrome oxidase by cyanide, C. Hexokinase by glucose 6-phosphate, and D. Carbonic anhydrase by carbon dioxide. So, here this is the question related to inhibition of the enzymes, that is competitive inhibition. So, here competitive inhibition means an inhibitor which is having the similar structure to that of the substrate molecule will inhibit the activity of the enzyme. How does it is doing means, here the inhibitor, competitive inhibitor, will go and occupy the active site present on enzyme molecule. So, it will occupy the active site on the enzyme molecule before the substrate will go and occupy the active site, such that now when the substrate molecule will come and try to occupy the active site, now the active site present on enzyme is already occupied by the non-competitive inhibitor. How does it is possible? Because there is a relationship between the active site shape and structure with that of the shape and structure of the substrate molecule. So, despite of this one, how does a non-competitive inhibitor will go and occupy the active site means, because the non-competitive inhibitor will exactly having the same type of structure as like that of the substrate molecule. That is the reason why it can easily go and fit into the active site of the enzyme. So, here malonic acid or malonate is an competitive inhibitor for succinic acid or succinate. That is, this is a substrate molecule and this is a competitive inhibitor. Actually, succinic acid should go and occupy the active site on an enzyme called as succinic dehydrogenase enzyme. This is an enzyme. But before the succinic acid will go and occupy the active site on succinic dehydrogenase enzyme, the malonic acid which is having the same structure as that of succinic acid will go and occupy the active site on the succinic dehydrogenase enzyme. That is the way how this malonic acid is an competitive inhibitor for succinic acid and occupying the active site on succinic dehydrogenase enzyme. So, from the given options, option A is the correct answer. Question number 11. The lock and key model of enzyme action illustrates that a particular enzyme molecule, options A, may be destroyed and resynthesized several times, B, interact with a specific type of substrate molecule, C, reacts at identical rates under all conditions, D, forms a permanent enzyme substrate complex. So, here lock and key hypothesis which was given by Emil Fischer and Koshland. So, here Emil Fischer had given this concept where there is a relationship between the lock and key, that is exactly the levers which are present in on the key will have the similar type of the insertions, so that it can open that specific lock only. That is what the relationship that is existing between the shape of the substrate and with that of the shape of the active site present on the enzyme molecule. So, here the relationship that is existing between the lock and key and the enzyme and substrate are they will interact with a specific type of substrate molecule only. Why we are calling that as a specific type of substrate molecule means, if any other type of substrate will try to occupy the space active site on the enzyme molecule, that substrate will not exactly fit into the active site. Why? Because the shape of the active site is different from that other type of substrate molecule. So here the interaction will be same, and here whatever the intermediate or shape that is formed here, that is whenever a specific type of substrate will come and occupy the active site present on the enzyme molecule, they form an temporary structure that is called as a temporary enzyme substrate complex will be formed. So it is not a permanent structure, and here this enzyme substrate relation is that was illustrated by lock and key hypothesis. It is not saying about the destroyed and resynthesis of several times, and here that is the identical rates of all conditions is also not related with the lock and key hypothesis. So from the given options, option B, where interact with a specific type of substrate molecule, is a correct option. That is, option B is a correct answer. Question number 12. Michaelis-Menten constant Km of an enzyme is substrate concentration at which the reaction attains. Options: A, its maximum velocity, B, half its maximum velocity, C, double as maximum velocity, D, its normal velocity. So here the Michaelis-Menten, that is Menten constant, that is Km, talks about here the half the maximum velocity. That is if you put the rate of reaction on a graph, on the substrate on one side and rate of reaction on the other side, here starting from here, wherever the substrate is consumed, the rate of reaction will be taking place like this, but the maximum velocity that is attained will be here, whereas according to Michaelis-Menten constant, half the maximum velocity is considered as a Km value. So based on the graph that have been taken, used by here in the Michaelis-Menten constant, so here the Km of an enzyme is a substrate concentration at which the rate of reaction have been attained, it's half the maximum velocity. So from the given options, option B is the correct answer. Question number 13. A ribosome is a, options: A, an enzyme associated with ribosome, B, an catalytic RNA, C, an enzyme that helps in ribose synthesis, D, an enzyme that joins ribose with adenine. So here ribosome, actually enzymes are catalytic in their nature. Those enzymes are proteinaceous in their nature. Actually all enzymes are proteinaceous, that is they are having the protein part, but here ribosome is an enzyme which is not the proteinaceous in its nature, but it is a RNA molecule which is having the catalytic ability or catalytic property. So from the given options we can say that ribosome is an catalytic RNA molecule, that is which is helping in the catalysis of the reactions as like that of the other enzymes. So from the given options, option B is the correct answer. Question number 14. 3 of the following statements about enzymes are correct and 1 is wrong. Which one is wrong? Options: A, enzymes required optimum pH for maximal activity. B, enzymes are denatured at high temperature, but in certain exceptional organisms they are effective even at temperatures 80 to 90 degrees centigrade. C, most enzymes are proteins but some are lipids. D, enzymes are highly specific. So let us start with the first option, that is enzymes required optimum pH for maximal activity. So here some of the enzymes are active at acidic pH, some of them are active at neutral pH, and some of them are active at alkaline pH also. So here the pH at which they are active, that is what we call it as optimum range. So all the enzymes does not have the same optimal range of pH activity, so they have different optimal activity. But here one thing to be noted, that enzymes require optimum pH for maximum activity. So statement A is correct. Statement B, enzymes are denatured at high temperatures, but in certain exceptional cases they are active at temperatures of 80 to 90 degrees centigrade also. Yes, that is correct, where all enzymes we can say are proteins. Here at high temperatures the proteins will become denatured, that is they lose their nature and they will not act as a catalytic in their nature. They will lose the catalytic nature, simply to say. Here when we consider an organism which is present, which have the ability to grow in high temperatures, so thermophilic we can consider. In those organisms the enzymes are present which have the ability to sustain and active at high temperatures also. That is the exceptional cases. So we can say that that statement is also correct. When it coming to option C, that is most enzymes are proteins but some are lipids. So almost all the enzymes are proteins in their nature, none of the enzymes are lipid in their nature. So all enzymes are proteins. So in the given option that is wrong. And D, all are, option D, enzymes are highly specific. Yes, that is correct. Why? Because if you consider an enzyme, that enzyme will act on that particular type of substrate molecule only, and if you take the same enzyme for other reaction where it have to act on the other substrate, it will not show any type of catalytic activity in that reaction. That is, one enzyme is there that is specific for that particular type of substrate molecule only. So that is, enzymes are highly specific. So this statement is correct. So in the question 4 statements have been given, out of that 3 are correct and 1 is wrong. So we have to identify the wrong one. So the wrong statement in that one is C. So the correct answer is option C. Question number 15. Which of the following statements regarding enzyme inhibition is correct? Options: A, competitive inhibition is seen when a substrate competes with inhibitor to occupy other than active site. Option B, competitive inhibitor is seen when the substrate and the inhibitor compete for the active site on the enzyme. Option C, non-competitive inhibition of an enzyme can be overcome by adding large amount of substrate. Option D, non-competitive inhibitors often bind to the enzyme irreversibly. So here this is the question related to the enzyme inhibition. So are enzyme inhibitors. So already we know that basically 3 types are there: competitive inhibitors, non-competitive inhibitors and the feedback inhibitors. So here the competitive inhibitors are the one which have the similar structure as like that of the substrate molecule, and they will occupy the same active site on the same enzyme. That is the competitive inhibitor seen on the substrate which will inhibit or which will compete for the active site present on the enzyme molecule. Whereas non-competitive inhibitor or inhibition. So the non-competitive inhibitor is the one which does not have the similar structure as like that of the substrate molecule, and the non-competitive inhibitor will occupy any other site on the enzyme molecule other than the active site, but it will, whenever it get attacking the other site, it will show its effect where it is changing its shape of the active site. So that is a way how the non-competitive inhibitor, despite of not having the same structure and shape as like that of the substrate, but it is inhibiting the enzyme activity. So this is a way how we can say that option B, where competitive inhibitor is seen when the substrate and the inhibitor compete for the active site on the enzyme. Why? Because the competent inhibitor and the substrate both have the similar structure and both are equally will be competing for the active site present on the enzyme molecule. So, from the given statement, statement B, that is option B is the correct answer. Question number 16. The enzymes enhance the rate of chemical reaction by options A. Increasing reaction time, B. Decreasing reaction time, C. Reducing activation energy, D. Increasing activation energy. So, here the substrate and the products are there, that is substrate will be converted into the product. In absence of the enzyme molecule, the substrate will be converted into product and it have to pass through an energy barrier. That energy barrier is called as a activation energy. It may be either in the endothermic reactions or the exothermic reactions, where in the endothermic reactions the energy will be utilized, whereas in the exothermic reactions the energy will be released. So, here if you plot a graph, consider the energy levels here. This is energy level present in substrate molecule where it had to pass through an energy barrier to convert into the product in absence of enzyme molecule. Whereas here we can say that this is energy level of the substrate molecule and this is energy level of the product molecule. That is, substrate will have more energy here and product will have less energy here. That is, energy will be released when the substrate is converted into product, that is exothermic reaction. That is in absence of energy, that is in absence of enzyme molecule. The same product is, the same substrate is taking part in the reaction in absence, in presence of enzyme molecule, where it is given as to same type of product with the same amount of energy. But here the energy barrier have been decreased. So, that is in presence of enzyme. That is, here this amount of energy is decreased by the enzyme molecule, so that the substrate is easily converted into, that is not, we can say that it can be converted into product in a short period of time. That is, the enzyme will decrease the activation energy such that it will fastens the reaction rate of reaction. So, from the given options, the enzyme will enhance the rate of chemical reaction by decreasing the activation energy, that is the energy barrier, that is transition state we can say. Energy barrier will be reduced or decreased by the enzyme molecule. So, from the given options, option C, that is reducing the activation energy, is the correct option. Question number 17. Feedback inhibition of an enzyme is influenced by. Given options are A. Enzyme, B. Substrate, C. End product, D. External factors. So, this is also the question related to the inhibition, that is feedback inhibition. That is, feedback inhibition means that is inhibition of the enzyme process because of accumulation of end product. So, let us take one example here. Glucose, by taking an ATP molecule, it will be converted into the product, that is glucose 6-phosphate and ADP will be formed. Here the enzyme which is taking part is a hexokinase enzyme. This is the end product. So, when this end product is accumulated, this will show its effect on the enzyme, that is hexokinase. So, that is the product is showing its activity on the hexokinase. So, the activity of that enzyme will be decreased. When the activity of this enzyme is decreased, it cannot participate in the reaction where the glucose and the ATP are the substrate molecules. That is the way how the excess or accumulation of the end product is showing its effect on the enzyme which is taking part in the first reaction. This type of inhibition is called as a feedback inhibition of the enzymes. So here feedback inhibition of the enzyme is influenced by the end product. So from the given options, option C, that is end product, is the correct answer.